In our today's lecture, we will be starting our new chapter, that is the cellular radiobiology, and the topic that we are going to cover is target theory. So before discussing about the target theory, we will first discuss about the basis of the target theory, and according to it, the cell contains many species of molecules, and most of which exist in overabundance, as you can see in this diagram. And radiation to such molecules would not result in noticeable injury to the cell, because there will be other similar molecules available to continue to support the cell. But on the other hand, some molecules in the cell are considered to be particularly necessary for normal cell function, and these molecules are not therefore abundant, and they are very important for the normal cell function. So radiation damage to such molecule can result in the severe damage of the cell, and there will be no similar molecules available as substitute. So this is the concept of the target theory, and according to it, for a cell to die after the radiation exposure, the target molecule must be inactivated. And this figure is explaining the whole target theory, the basis of whole target theory. You can see a cell here, and you can see that when the target molecule is not irradiated, which is in this case is DNA, and other molecules that are present in abundance are irradiated by the radiations, there will be other molecules present to work as substitute for those molecules. But if the target molecule is the DNA and it is being irradiated by the radiations, then the cell death can result, because this is the only or we can say that the major functional molecule to work for the cell, and there will be no substitute available. So ultimately the cell death will result. The next very important point that we are going to discuss is the hit. But before that, just remember that the target theory was previously used to describe the lethality of the cell, but now we use it as intracellular molecular cell death as well. That is for the known lethality of the cell abnormalities caused by the radiations. The next is we will talk about the hits, but before that we will discuss about the target area as well. And you already know that the target area is considered to be an area of the cell that is occupied by the target molecule or by a sensitive site on the target molecule. So this area will change because of the intracellular molecular movement, and the radiation interaction with these cellular components would be random. So the interaction that will happen between the radiation and the target molecule, that will also be random, and there will be no favoritism by the radiations. So the next that we are going to discuss is the hits. So what basically the hits is? Hit occurs when the radiation interact with the target. So we say that a hit is said to have occurred when the radiation interact with the target, and these radiation can occur not only with the target molecule, but they can also interact with the other molecules as well. So hit is not necessarily can occur after the DNA and radiation interaction. It can also occur with the other target molecules that are present in abundance. We will say that in that case hit has also occurred. Another important thing is the phenomena, we can say, through which the hits occur, and it can occur either with the direct effect or with the indirect effect. So the first phenomena that we are going to discuss is by the direct effect. This image is very important, and before going into detail, we will first talk about the linear energy transfer. That is the rate at which the radiation is transferred to the soft tissue. So this is basically a rate at which the ionization radiation is transferred to the soft tissue. And there are two factors responsible for the direct and indirect effect of hit, we can say, and these factors are linear energy transfer and the oxygen effect that we have previously discussed. So what basically happened according to this diagram, according to the first image, there is low linear energy transfer radiation presented, and you can see there is a large difference between the molecules. So the probability of a hit on the target molecule would be low because of the large distances between these ionizing events. In the second scenario, this is in the presence of oxygen, and there will be free radicals formed, as we have discussed in the radiolysis of water. So there will be free radicals available, and they will form, and the volume of effectiveness surrounding each ionization will be enlarged, and consequently there will be probability of a hit, and there will be increased probability of a hit. In the third scenario, we have high linear energy transfer radiation, and you can see the distance between the ionization is so close, so the probability of a hit would be direct, and that probability of interaction through the direct effect is very high. The last scenario is when the oxygen is added to the system with the high linear energy transfer radiation, and in this case the maximum number of hits has already been produced, so there will be no additional hits result. So that's all about the cell theory, and we will summarize it. First, we talked about the basis of the target theory, and we studied that there are some molecules in the cell that will be in abundance, and if the interaction occur between the radiation and those molecules that are abundant, then there will be no cell damage because there will be substitute available for those molecules. So there will be ultimately no result or no effect. In the second case, if the target is, for example, the DNA, that is the only functional molecule, then definitely there will be no substitute available, and it will not present in abundance, and damage to the DNA will result in the cell death. So this is the basis of target theory, and then we studied that the target theory says that for the cell death, the target molecule has to be inactivated, and the DNA in that case is a target molecule. Then we talked about the application or usage of the target theory, and we said that we originally used that for to represent the cell lethality, but it can also be used to describe the non-lethal radiation induced cell abnormalities. Then we talked about the target area, and we said that this is the target is considered to be an area of the cell that is occupied by the target molecule or by a sensitive site of the target molecule. And the next point that we studied was that the interaction of radiation and the target molecule will be random, and there will be no favoritism by the radiations. Next we talked about the hits, and first we described that the hit is basically the radiation interaction with the molecules, and it can be it's not necessary that the interaction will be always between the target and the radiation. It can happen with other molecules other than the target molecules, but it is not possible for us to distinguish between the direct and indirect hit. And further we talked about the direct and indirect hits as well, and we studied that there are basically two factors that are responsible for hits, or there are two phenomena by which the hits basically occur, and it also depends on the linear energy transfer and the oxygen effect. And then we explained some scenarios through this diagram. In the first scenario, we said that we have low linear energy transfer radiation, and we also described the linear energy transfer, that it was the rate at which the ionizing radiation are basically transferring to the soft tissue. So in the first scenario, when the low LET radiation are used, in the absence of the oxygen, the probability of hit on the target molecule will be low because of the relatively large distance between the ionizing events. The next is in the presence of the oxygen, and because of that there will be the formation of the free radicals, and the volume of effectiveness surrounding the each ionization will be enlarged, and consequently the probability of a hit will also increase. In our third scenario, we said that we have high linear energy transfer radiation, and the distance between interactions is so close that the probability of a hit would be through the direct effect. Then in the final scenario, we said that we have added oxygen to the system in the presence of high linear energy transfer radiations, and the added sphere of influence for each ionizing event will be larger, and there will be no additional hits result, because the maximum number of hits has already been produced by the direct effect with the high linear energy transfer radiation. So that's all about the target theory, and if you have any question regarding this, then you can ask, and that's all for today. Thank you so much.